Okay, we are now live on YouTube. So welcome to the third day of IWS series hosted by Department of Biotechnology and Bioinformatics, Kuvempu University. Today we have session 4 on UALCAN, that is a comprehensive anti-integrated cancer analysis tool, which will be presented by Dr. Darshan S C. So today we are going vocal for local because he is our own local guy from Shimoga, and he is a very hardworking guy. He was a student of my first batch when I joined Kuvempu University. So I got very little time to interact with them, only probably 4 months. But I found him very highly hardworking and striving always for the excellence. So he completed his MSc in bioinformatics from our university in the year 2007. And then later he joined the lab of Dr. Kshitish Acharya at IBAB. And since then, he worked on the developer side of bioinformatics. So he was very highly motivated, and he was very much involved in cataloging the genes which are important for tissue specific expression. So the other day, Dr. Kshitish Acharya told you about this guy, hope you remember. And he actually registered for his PhD at Mahe, now which is currently Manipal University. And he did his work at Kshitish Acharya lab and got a PhD from Mahe, Manipal in the year 2015. Then he went to, he is currently working as a postdoctoral fellow since 2015 at Surya Narayana Varamballi's lab at University of Alabama at Birmingham, USA. He has 33 publications in peer-reviewed journal to his credit. And those 33 publications have accumulated more than 1,086 citations as of today. So that is a testament that what kind of quality research and publication he has done in his field and how much he has contributed to the research. And he is very much, expert in in-depth analysis of data from microarray and various next generation sequencing technologies as RNA sequencing, chip sequencing, and whole exome sequencing. And he is very good at developing a user-friendly web portal and analysis platform. Some of them are Green, Megex TDB, Megex UDB, and UALCAN. So the UALCAN server is the recently developed server which went on air 2 years ago, and it recorded a very highest number of visits within 1 month of its launch. So it's very user friendly, and it is going to give you a platform for comprehensive analysis of expression in different types of cancer. And using that, several people have quoted that particular server and in their publication. So that is a testament that how user friendly is the server. So the first contribution of Dr. Darshan was development of Green repository, that is a genomic repeat element analyzer for mammals. So that is what he developed when he was working at Kshitish Acharya's lab. Then currently he is he has developed UALCAN, and he is developing much more servers, I presume. He contributed to the characterization of glycine N acetyltransferase, and he published that also. And he also contributed for identification and analysis of expression and role of methylene tetrahydrofolate dehydrogenase 1 like in bladder cancer, and characterization of glycine N acetyl transferase like GLYATL1 in prostate cancer. So though it appears like like the um, uh, previous two speakers had contributed more to the cancer, like Dr. Darshan and Dr. His mentor, Dr. Kshitish Acharya. But one is at the user side, and our Darshan is on the developer side. So that's the two facets of development of any any repository. So you will be having either the user side or the developer side. So he is very good at programming skills and developing the web interfaces and several servers. So with this very small introduction, there is a whole lot of thing which is available on his Google Scholar as well as his university website. But I don't want to bore you with all those things, but I will share you the link. You can go and you can watch yourself about his achievements. And he is one guy whom I usually quote in my class as to, like, you don't need to know everything when you go out there to work, but only when you have enough amount of zeal to learn. The same thing the other day Dr. Kshitish Acharya also said, that if there is a zeal to learn and mentality to put everything into whatever the work you are doing, so you can really excel in your field. So Dr. Darshan, I'm very happy that Dr. Darshan is an example for that statement by Kshitish Acharya, and also I endorse it completely. So with this few introductory remark, I welcome Dr. Darshan, and I request him to uh, take the session and deliver his lecture.
Thank you, sir. It's it's not a short; it's quite an elaborate introduction, and thank you for that elaborate introduction. So, shall I share my screen now?
Yes, you can. You can go and you can share the screen. Yes.
Hope all of you can see.
Okay. Fine. It is visible.
Yeah. So, before first of all, good morning to all. Uh, Before I begin my presentation, I would like to thank the organizers for giving me such a wonderful platform to share my research. And to be frank, I'm extremely delighted to share my work to the students, the faculty of the same department from where I spent my 2 precious years and graduated with a master's degree. And with that, this is one of the snapshots which I have from our farewell ceremony, to be frank, in 2007. So, yeah.
Nice to see you still have that photo.
Yes,
My
sir,
God.
I do have that. So yeah, it's one of the important days. So definitely yes. So with that, I'll start my presentation. So today I'm going to talk about. I hope I'm quite audible to all of you. Sir, is it fine now? Okay. So uh, today I'm going to talk about a web server that was developed in our lab at the University of Alabama at Birmingham. So I joined the lab as Sir told, in 2015, and one of the initial projects that I took up was the cancer data analysis. I was new to the cancer field. I worked mostly on the reproductive biology side. Again, data analysis itself, but still. So this was the first project that I took up in 2015, and within a couple of years we developed it. So today I'm going to divide my entire talk into 4 major sections. So I'll begin by listing factors which actually motivated us to develop this particular web server. And then I will list the steps that we took to develop this tool. And I'll briefly demonstrate how to use this particular tool, uh, to how to maximize its features uh, with the perspective of a clinician or a cancer researcher. And finally, I'll touch upon a couple of points about what are the future plans that we have regarding this particular research. So let me begin with the factors that motivated us. As most of you are aware, that in the age of high throughput experiments like microarray, sequencing, and mass spec, multiple laboratories are collaborating and they're working towards single goal, that is to understand the cancer better so that we can end up with a better therapeutic uh, methods, better therapeutic targets. So there are multiple projects like that. So some of them are like the Cancer Genome Atlas program, which started in 2006 and ended in 2015. This is an initiative from National Cancer Center, USA. And we also have something called the International Cancer Genome Consortium, which aimed at sequencing the genomes of the untreated cancers. Uh, Also have something called a target, which is the full form for the therapeutically applicable research to generate effective treatments. And this particular program is mainly directed towards understanding childhood cancers. So other than this, there are multiple projects that are currently undergoing, and they're unearthing new discoveries. So when I begin with the cancer data analysis, one of the first project that stuck to my mind that time was the Cancer Genome Atlas program, because it was about to end in 2015-16, and they were releasing the data for the public to use. So briefly about the Cancer Genome Atlas program. So this is a program which was conducted by at least 20 laboratories from US and Canada, where they collected the tumor and normal samples from approximately 11,000 patients corresponding to 33 different types of cancers. Their objective was to use all modern technologies like microarray, sequencing, the whole RNA-seq, whole exome sequencing, and even the proteomic technologies to understand each and every molecular behavior in 11,000 patients. Thereby, they wanted to understand the molecular basis of cancer. They want to unearth new types of tumors, new tumor subtypes. They want to also identify some of the new biomarkers which they can target to treat cancer. So the with the efficient work of 10 years, they were able to publish around 30 to 40 papers in from 2015 to 2017 in many reputed journals like Cell, Nature, and others. And with all this data and information available, the first thing that after going through all this, the two questions that I addressed our lab was, uh, is there any scope to dissect this data further? It's a huge data. As you can imagine, you are looking at about 11,000 cancer patients and looking at every bit of molecules from gene, protein, from microRNA, and all other types of molecules that you're looking at. And is there any scope to further dissect this further was one of the questions. The second question is, how easy for anyone who is programmatically limited to access this data. So because as you can imagine about the volume of the data that is generated, a typical researcher with a typical computer or a laptop, can they look at this data and make sense out of it other than going to the paper? So those are the two questions that I initially had. As far as the first question is concerned, we quickly realized that there's definitely a very good scope to dissect this data further beyond what they have already published. One of the reason being, even though in the last decade, there is lots of new technological revolution and lot more biomarkers are reported. As far as protein research is concerned, 75% of the protein research still work on 10% of the proteins. So they still work on the same P53 protein, KRAS proteins related to cancer, and try to understand its involvement and how can they come up with a better treatment methods. And the second thing is there is a review which was published in 2017 now. So this actually is from MD Anderson Cancer Center, one of the most reputed cancer hospital in USA, as far, and even the world, to be frank. And they are specialized in precision medicine. So one of the study from the University of Texas tells that about 739 patients enrolled for the precision medicine. But after genetic testing, only 16 of them were able to get the precision medicine. So basically, they tell that most of the cancer patients currently do not benefit from the current precision medicine, and there is a definite need to find new targets. So definitely there is a requirement to mine this TCGA data further to get new targets. So with this in mind, we thought of starting the projects, but initial thing that we wanted to look at was actually the volume. As I told, it's a huge data. The huge data is this much. So you have about 2.5 petabytes of data from TCGA generation, which equates to about 500,000 DVDs. That's a lot of data for even not even the bigger lab, smaller lab, even for the bigger lab, and for most of the people, it's quite a lot of data to deal with. But thankfully, TCGA provides information in basically three levels. Level one is mainly raw data. Raw data is the data that comes out of a machine. So any sequencing machine or microarray chip image. So all those information what they get, so that's a raw data. The level two is basically a normalized data, which which is slightly smaller in size compared to raw data. And level three is actually something we call it as a segmented data or annotated data. This particular data is quite small in volume. I mean, basically, the entire 2.5 petadata in that it constitutes only about 2 to 3 GB. So supposedly pretty small in size and can be handled better with a computational programming. So we thought we'll start with the annotated data first and then try to move our way upwards. So before we begin the project, we wanted to make a survey in 2015. Are there any tools which have already did this? They have used this segmented data or level 3 data and developed a portal. So when we checked at that time, there were many. So these were the most famous at that time. So CBio portal, I believe some of you might have used this. Tandric, Firebrowse, Mirgator, IsoExpress, for each of them were developed for different objectives. And they were, because as I told, the TCGA provides information about protein coding genes, link RNA gene, microRNA genes. So each of, and mutation, methylation. So each of them actually directed towards different aspects. And after careful studying of all these tools, we came to the conclusion that definitely there's a need for a new tool which actually helps user to analyze gene expression and patient survival in tumor subgroups. So I will explain more in detail about the survival thing, but the main aspect is these tools don't allow efficient analysis of the tumor subgroups. It will tell you exactly which genes are highly expressed in breast cancer or a prostate cancer, but it will not help you to study it in depth in terms of stage, in terms of race, in terms of gender. So all this information one cannot study effectively in these platforms. And that is what prompted us to develop a new. So with that in mind, we began the development. The first stage, as I mentioned, uh, of the development was acquiring it, collecting the information. So TCGA actually has all the data available. Anybody in the world can access the data from a portal called as a GDC, Genomic Data Commons. So anyone can go there, and it's a government, US government website, because it's funded by US government. They have put in their website, and anyone can access the data. So there are multiple tools available. One of the R-based tools called as TCGA assembler. I use the tool to download the data. And when I download the data, it's basically text file. It will have a simple text file, which is very big in size, lots of numbers, and each cancer you will have one particular file for one particular type of information. For gene expression separately, for mutation separately, methylation separately, but you have just simple text file. So the first objective was to organize this data and categorize it further. As I mentioned, we are not interested in looking at entire cancer. We are mainly interested in looking at subgroups of cancer. So I used in-house R and Perl scripts to organize this data. Just for example, let me tell you how I did with the bladder cancer data set. So in case of bladder cancer data set, they had gene expression information for about 408 patients. And the initial task was to organize this data and categorize it based on different clinical features and demographic features. So one of them was the race based. So this 408 was divided into 320 for the Caucasian race, 22 for African American, and 44 for Asian. So similarly, the same approach was applied to do as for the stage based and gender based and more different types. So I'm just giving an example how it was done. So after collecting data, we segregate the data, and eventually we ended up with the host of files, lot of files for uh, 33 cancer types, files corresponding to RNA-seq, microRNA sequencing, and DNA methylation data. And recently, in last 1 year, we have done it same thing for proteomic data. So this was collected from another consortium called as CPTAC, Clinical Proteomic Tumor Analysis Consortium. So they provide information for 7 different cancer types. So all this actually constitute the genomic information, gene expression, mutation information, methylation information for approximately 20,500 proteins and 1,870 microRNAs in 53 different types of cancers. Uh, you can just imagine about the dimension. So I'm talking about for each gene, we have information now for in 33 different types of cancers. And same thing goes with the proteins. So recently acquired data, which constitutes about the gene expression, the protein expression information for approximately 10,000 proteins in 7 cancer types. And all these information that we gathered and categorized was subjected to statistical analysis and using R statistical packages. And all those analysis results were actually we wanted to post in a website. And the website was actually developed using Perl Apache interface, Perl CGI interface on Apache web server, and all the graphics were actually designed by using the JavaScript's hijinks. So that is, in simple words, that's how the server was set up. Currently, this is actually a snapshot of the UALCAN website, uh, and you can see the the URL, which is currently, or you can just Google UALCAN. I'm sure it's the first or second search result in the Google website. Server. So currently, I mean, for those who are interested in computer stuff, especially the hardware things, so currently we the entire UALCAN website is hosted in a Dell PowerEdge server with the capacity of about 22 TB hard drive space, 100 GB RAM, around 72 CPU cores are used, and it's running on an Intel Xeon processor. So the operating system that I am more acquainted with is Linux, and definitely CentOS is one of my favorites. So that's why I use this, and it's also light. So currently we also have a backup server with the same capacity for this particular tool, and that's the actual story of the UALCAN development. So, so now I'll briefly describe or demonstrate how to use UALCAN. Actually, the UALCAN name itself was designed in such a way that my mentor wanted the site to be as user friendly so that he can tell that you all can easily use the tool for all the clinicians that are out there in UAB or his friends and all. So it was designed in such a way that it should be very easy to use, and it is targeted towards especially those clinical researchers, those cancer researchers who are not equipped with programming skills. So they should be able to easily click the buttons and get the results and interpret it easily. That's the objective of designing and developing this program. So this is a snapshot of the analysis page. So UALCAN currently has 3 panels for any user to use to actually query that database, internal database. So each panel is designed to provide different type of output based on the user. The panel 1 is mainly designed to any new user who is like less involved in, or they are involving initially in cancer research and all. So it's mainly designed to list top overexpressed and underexpressed genes in different cancers. So here we, for example, I'm just showing an example of a breast cancer. So when a user clicks on that particular link, they are directed towards a page which shows a heat map. Heat map of, in this example, I'm showing 25, but together it shows user, it allows user to see about 250 overexpressed and 250 underexpressed genes in particular cancer.
Excuse me, Dr. Darshan,
Yes,
excuse me.
yes, yes.
Could you please, for the benefit of the student, could you please tell them what exactly is a heat map?
Okay. So heat map basically is a graphical representation of the expression value. So, okay, let me tell you that in case of breast cancer, there are the TCGA has uh, samples for about 114 normal tissue and about 1095 tumor tissues. Okay, so the heat map basically shows the level of expression of a particular gene in each of those individuals. So what you can see here is initially the one block where you can see is this is the expression of a particular gene called 10A1 gene in 140 normal tissues, and this is the expression of uh, the call 10A gene in 1095 tumor tissue. So each line you can see here is colored differently because it shows the level of expression. Lower the value of expression, darker the color. So you can see that the call 10A1 is very has a very low expression in normal patients or normal tissues, whereas the same gene is highly expressed in the majority of the tumor tissues. So you cannot tell that all it is highly expressed in all the patients, but definitely it is highly expressed in majority of the tumor samples. So that is what is depicted in the heat map here. So each line, which is very closely associated or located to each other, they represent one value here. So eventually the level 3 data, what I was referring to earlier, basically bits of values. Now you have, for each gene, you have value from each patient. So what I'm telling, I'm replicating that entire values in a graphical format so user can easily see this and visualize it in a better way. So it's a way of showing, it's one of the most common ways of showing top genes in different conditions actually. So user can download this information as an image, and most of them directly use this in their publication and use for the further analysis. And again, I'm mentioning, this feature is mainly for those who are new to the cancer and they wanted to identify genes that are highly expressed or low expressed in cancer compared to normal. And this feature is available only to about 14 out of 33 cancer types, because for rest of them, there are either the normal samples are not sequenced, or they have a very low number of normal samples, something like less than 10 normal samples. So uh, it doesn't make a lot of sense to look at something like 300 tumor samples versus 2 normal samples. So we don't make that kind of a comparison. So only those cancers which have at least 10 or more normal samples we consider and provide this particular feature. I hope that is clear now. Uh, So the next panel is basically designed for those users who are looking to extract information for the gene of their interest in specific cancer. So I'll just explain this with an example of gene called CDC25C. It's a cell cycle related protein, and I just, with the intention of obtaining details about this gene from related to breast cancer, how one should go about it. So one can enter the gene symbol in the text box, select the cancer of interest in the drop-down menu, and when you click submit, you will be directed to an intermediate page. And this particular page has different links. Uh, and each of these links, you can see the expression, survival, methylation, correlation, and pan cancer, when clicked, will direct user to different pages and provide different types of information. Suppose a user clicks on expression. He or she will be directed to a page where user can see the bar plot, which initially shows the relative expression of the gene of interest in breast cancer compared to normal. So you can see that the CDC25 is highly expressed in breast cancer compared to normal. We also provide the statistical significance, which is calculated using unpaired t-test. And as I mentioned earlier, we were more interested in tumor subgroups. Uh, any plot that I am showing in this during this presentation can be downloaded as a publication quality image or a PDF from the website. So user can click on that button and they can download the information. So in addition to the overall expression of tumor versus normal, we provide the tumor subgroup information. So by changing option in this drop-down menu that is available just above the box plot, user can see the expression of level of the same gene in different stages of the cancer, different races, based on age groups, based on subclasses, based on TNBC subclasses, menopause status. So these are a few examples. These information varies from cancer to cancer. So in case of breast cancer, these are the different plots that one can obtain, and we also provide statistics for each of the plot. So when a user clicks on the show statistics, they will the statistic will be provided for normal versus the class 1, normal versus class 2, 3, like that. So they can decide whether it's indeed statistically significant or not. So the next aspect is the survival analysis. So I'm not sure that most of you are aware of this. In case of cancer research, the survival plot is one of the major contributor in identifying the biomarkers. Uh, Gone are the days where people just look at whether the gene is highly expressed or low expressed to decide it's important or not. So in addition to expression, nowadays people do look at survival plots. So how, before showing you the results of the UALCAN, I thought I'll just briefly describe how the survival plots are derived. I hope it's not a repetition. You might, I don't know whether you've already been taught about this. Uh, so if it is true, then I will move on, but I'll briefly try to explain. So what happens is, as I mentioned earlier, when I download the information, we do get something called the clinical data along with all those experimental data. We get the clinical data. So the clinical data looks like something like this, as you can see on the screen. So this is an example of a clinical data for about 16 patients. So where you get survival information. For example, the row 1 indicates that the patient 1 who is diagnosed about 23 days ago with cancer is still alive, and the level of expression of the gene X is 189. 189 is the expression value of gene X. The second row indicates the patient 2 who is diagnosed of cancer in 123 days back is not alive yet, so he's dead. And the expression level of the gene is 380. So like that, you obtain this clinical data, and this is the gene expression code. So we combine both of them together to obtain the survival plot. So the first step involved here is, first you will do is sort the patients based on the expression level. So this is what I have initially, and I sort the patient data based on the level of expression of gene X. So what happens now, you can see that the patients are sorted based on the gene X. So I can divide this entire list of patients into two groups: patients with low expression of gene X, patients with high expression of gene X. This is actually a dummy example. So you can clearly see here that patients who show high expression of gene X are more often dead than not, whereas patients who have low expression of gene X are more alive. So basically, high expression of gene X is dangerous to the survival of the patients. And that is what you want to understand from the survival plot. So this entire data, what you see on the screen, is represented as a plot. We call it as a Kaplan-Meier plot. So this is how it is shown. So you can see that the survival rate of the low expression is pretty high, whereas survival rate of the high expression, which is a red line, is pretty low. So the difference between the survival rate of low expression and high expression is estimated, and the significance is determined by a test called as log rank test, and that provides a p-value, which is what we use as a threshold. So basically, based on that, we decide whether the expression of a particular gene is detrimental to the health of the cancer patients or not. So this is just an introduction. So basically, for a gene of our interest, so we do provide that information in UALCAN. One can download. And there are two types of survival analysis that one can do. We call it as a univariate survival analysis, where we only consider the expression in the level of expression of a gene impact on the patient survival. And there is also something called as a multivariate survival analysis, where we look at not only the level of expression, we also look at other factors like cancer stage or cancer type or race and other features. So hope I explained it in a simpler way. Let me know if you have other questions later. You want me to explain it in a much simpler. So other than the survival, we have the methylation information. So TCGA provides methylation information from the microarray experiments. So they have something called Illumina microarray chips, which measure the level of DNA methylation in the genic and promoter region. So what we have done is, excuse me, so we have collected only those uh, probes corresponding to the promoter region, and we are providing the level of methylation, DNA methylation level in the promoter region of gene fragments. So this is again provided for all the genes that is available that are there in a microarray chip, and we consider only those probes that corresponds to the promoter region of the gene. So the next aspect that the UALCAN provides is actually the correlation information. So uh, many of you might be knowing that it's always interesting when you find a particular gene that shows high expression. And most of often the question is, what are the other partners of this gene? Is there any gene which behave in a similar way compared to the gene of interest? And the correlation analysis does allow you to do that. So we do the Pearson correlation analysis to list all the genes that are positively correlated with the gene of interest or negatively correlated with the gene of interest. And this information can be visualized as a scatter plot for any gene, and this can be again exported and used in the publication. So the final analysis view is actually the pan cancer view. This is again, it's little favorite of most of the uh, cancer researchers because they get to see exactly how the gene behaves in across all the cancers. So you can see that I've actually each cancer here is marked with 4-letter or 3-letter codes. So BLCA corresponds to bladder cancer, and these codes are actually given by TCGA. So I'm using the same codes. So at one shot, user can visualize my gene of interest is high in which cancers. What are the cancers in which it's really high, or it is showing opposite expression, or it shows no difference. So they can easily view this information in a pan cancer view, and again, this can be exported and used as per user's design. So in addition to all these information, I mean, it's very important nowadays that user wants something like a one-stop shop. They want all the information available at one place. They are not interested in going to some other website for some information. So what we have done is we actually provide links to the external database. The database are not located in UAD. They are very famous, most commonly used. So user can click on the links given in those places like HPRD, gene cards, target scanners. These are databases which are very useful for the cancer researchers. So they can click on those buttons and will be directed to the other databases and gather. So this is about the panel 2. Now the panel 3 is mainly designed for those researchers who work on particular pathways. Now, some people are very much focused. They always want to look at the data in terms of one particular pathway or one particular class of genes. So we have designed this particular panel for only those researchers. So what user can do is they can click on a drop-down menu and they will be shown a list of pathways which are most commonly associated with the cancer, and also list of gene classes like histone methyl transferase, DS which are involved in cell division and control, all those things. So user can basically select any of these pathways and they can query all the genes that are associated with this particular pathway. For example, I'm showing you a P53 signaling pathway, which is very well uh, associated with the cancer. So user will be presented with a huge table where each gene involved in that particular pathway is listed, and for each gene we provide a small a multitude of buttons where people, when they put a cursor on it, they will be shown a pop-up information whether that gene is upregulated or downregulated, or whether survival is significant or not. So user can see all the information at once, and they can click on any of these buttons to get to the gene expression and survival page and know more information about it, gather the graphics. So these are the three main panels that we have designed and targeted towards different audience. As I told initially, the panel one is for those who know little about the cancer. They just want to start the analysis with completely unbiased, in an unbiased fashion. Whereas panel 2 and panel 3 are for those experienced researchers who already have their pathway or gene of interest and want to explore more on those things. And this is something new. This was again, as I mentioned, last year with the collaboration of Dr. Chad Cretton from Baylor's College of Medicine. We integrated the proteomic data. Now, this is not available for all 33 cancers. And again, this is from the different set of patients completely. It's not from the TCGA, it's from the CP tag. So, but we do provide basically in one website, user can now study the RNA level as well as protein level expression of the gene. And we again provide this information for all the tumor subgroups that is available. So UALCAN was initially published in 2017 August in Neoplasia Journal. It was very much well received, and last year the protein data integration was reported in a Nature Communication paper. So this is from again the Baylor College of Medicine, and we are extremely glad that since, as Sadhu sir has mentioned in his introduction, it's pretty well used. Nearly we have about 400,000 visits since January 2017, and it's been used extensively from cancer researchers from all around the world, not only in US, and not only in China, but in many other places, Europe, in, even from India, we have a pretty decent number from Australia. So the simplicity of the tool, I believe that's what attracting the users, because you do, after our publication, we also found that many others have uh, created similar tools, but the simplicity of the tool and the ease with which user can download the information and use it in the publication may be made it popular. So we have over 500 citations for this particular tool right now. So most of them have been using this in the research and publishing with the figures downloaded from the. So as far as future plans are concerned, I mean, I'm actually working on the final stage of link RNA integration. So currently, link RNA is one of the key player in cancer biomarker discovery. So especially in case of diagnostic purposes. So lots of research are being done on long non-coding RNA, which is especially easy to detect the influx streams and all. And so we are gathering the information, and we've already gathered the information from TCGA and integrating that now to the UALCAN. And also want to expand the UALCAN data with the copy number alteration from the whole exome sequencing data from the TCGA and others. So that is the current plan as far as future plans that we are thinking. And yeah, this is just a snapshot of city where I live, Birmingham. It's southernmost part of America, in the state of Alabama, and this is the comprehensive cancer center where I work. There are about nearly 50 different PIs. The research, the main researchers are involved in working different types of cancers, ranging from prostate, breast, ovarian, skin cancers, and all. And it's located just adjacent to University Hospital. So basically, that's one of the features in USA where the universities and hospitals and all are together. So it basically, especially for the research purpose, the acquisition of the samples and all is a lot more easier in here. And we get it's UAB hospital is one of the reputed hospital in the southern part of America. So I'll complete my presentation by with acknowledgement. It's absolutely impossible to do it in a matter of 1 year, which we did the entire thing with the help without the help of all my colleagues who helped in acquisition and validation of the data. Because one thing that I would like to mention here is it's not about just downloading and doing the data analysis. So once you do the analysis, you need different pairs of eyes to look at the data and validate it. Many times, errors can creep up when you write a program, when you organize information, because if 1 or 2% is doing entire exercise, there could be errors, and we don't want any errors. With the help of many wet lab biologists like Dr. Chakra or Akshaya and others. So we were able to validate them. So basically, with the independent validators, we were able to validate whatever the analysis that we did. And we also, I'm indebted to my mentor, Dr. Suryanarayana Vadambal, who actually encouraged me to proceed with this. Initially, he was the one who used to bug me for the TCGA data, and uh, it's fascinating that when I joined the lab, he wanted TCGA data information for 1 gene, and the next day he wanted for 3 genes. So after 1 week, when I was trying to repeatedly doing the same thing, that's when this idea of UALCAN came. So we realized that instead of I doing the entire exercise for 1 gene at a time or few genes at a time, why not we develop a particular web portal where information is available for all the genes and he can access whenever he wants. And that's how this idea start uh, imploding, and we just did it in a matter of 1 year. We had the server ready, and we sent the paper for the publication. So thank you all for the patient listening, and please let me know any questions that you have regarding this or any general aspects. Thank you. Okay, the question that I have. from Hemavati is about how much difference in sequence between normal and cancer genome. So actually, that is to do more with a mutation. Uh, in terms of mutation, there definitely will be certain mutations that can creep up in the cancer genome, which might lead to the dysfunction of some of the proteins, and that could eventually lead to uh, maybe the hyper expression or hyperactivity of some of the oncogenes, or might suppress, in some cases, the mutations can also suppress the function of some of the tumor suppressors, the genes which are dedicated to prevent the tumors from happening and cancer from happening. So
Yeah.
that much could be there, but the mutation frequency actually varies from cancer to cancer. Not every cancer will have lots of mutations. So a cancer like, for example, the prostate cancer has a lot of gene fusions. So where the two different genes, they actually fuse and they express. We call TMPRS. There's a particular gene called TMPRS fuses and express. More than 50% of the prostate cancer, uh, nearly 50% of the prostate cancer patients have the gene fusions. Whereas in some other cancer, like colon cancer or rectal cancer, even in case of ovarian cancer, so there are lots of mutations. So mutations can actually creep up a lot in the tumors, or in the cells which are the tumors, and it can eventually lead. So it differs from cancer to cancer.
Okay, Darshan. So Dr. Manoj Kashyap is asking, are there plans to integrate RNA splicing data in near future, which are focused on RNA splicing event, alternative splicing events?
Yes, actually, that was one of the objectives, but it has one technical aspect or technical issue, I think. So the level 3 data do not have the transcript level information. So whatever that they release is more of a gene level information. So what we need to do is we should go to the level 2 data, that is normalized data, and then use and further process them to obtain the transit level information. So it is in our plans, but again, there are the complexities in terms of volume. So we need at least about probably about 20 to 25 TB of just a raw data to the process data to download and start the analysis. So it could be a PhD project for somebody else, I believe. So
Yeah.
that's a lot of work actually involved. Yeah, there's actually no tool available for that purpose.
Okay.
Yeah, one of the tool was developed earlier from Taiwan called as ISO Espresso. I mentioned in my presentation. It was working for some time, and then after 2 years, it's not. And that also they didn't have in-depth analysis, but they used to give some sort of transcript level information.
Okay. So one more question. I think the Dr. Anant Krishna is asking. I'll reframe it for you.
Mhm.
Out of hundreds of the up and down regulated genes
Mhm.
that you provide in your server,
Mhm.
so how will it help you to identify the biomarkers?
Well, first of all, it's a start. I mean, basically, one has to go through the entire list, and one way of narrowing it down is looking at both expression as well as survival, as I mentioned. So that will narrow it down further. Uh, the second thing is, again, you have to start with somewhere. So what people do is they will again look among them any pathway of their interest and all to further narrow it down and do it. So it is mainly something like a bird's view, because
I
instead of
think
you, yeah.
the person is going is the idea of the question is how
Mhm.
does a bioinformatician and clinician they sit together to carry out whatever the information is given to further validation. I think that's the idea of the question.
Okay. Well, it is a good question. It's a very good question, but it's not straightforward to answer. The thing
Okay.
is, uh, each of them have their own complexity. For example, some clinicians come to the bioinformaticians with open mind. They want to look at what is the best possible gene available based on statistics alone first. What are actually, if you give some 20 different candidates and then they go for the experimental things, then there are certain clinicians and researchers, they come up with a quite biased mind. So basically, when they enter to the conversation, they tell they're not interested in any other gene. Tell me about tyrosine proteins only, so something like kinases. A particular tyrosine kinases is what I'm looking at. Tell me whether these tyrosine kinases have or are candidate for the biomarkers. So again, it varies from person to person.
So, one candidate is asking, this is a vague question. Could you please let me know,
Mhm.
is it helpful in carrying out studies in vitro cell line studies? I mean, like what she might be asking is most of the data that is coming from in vitro cell line studies, how can you correlate it to the live clinical sample? I don't think you people will be taking the results of microarray from clinical sample, it seems, not from the cell line.
Yes. So in this case, well, again, it depends. This is a better data when you look at when you look at a clinical data. It is a lot more better than working on a cell line. Uh, but again, cell lines are also of different types. I mean, if I identify from UALCAN a biomarker for suppose oral cancer for a Caucasian male, suppose Caucasian male, if I identify using all the analysis, then I have to basically do the wet lab study. If I don't get a Caucasian male sample, I can actually get cell line which is derived from the Caucasian male and perform the analysis. So like that, we can validate and do it.
Okay. How can we identify the level of methylation? That's the next.
Yes. Yeah. So in the data that I showed, uh, the plots actually have something called a beta value. Now, from the microarray, what we gather, we obtain something called we derive the methylation level as a beta value, which ranges from 0 to 1. So, any probe which has a methylation value of zero point 0.5 or more will tell me that this particular region is hypermethylated. Any probe which has a beta value of 0.25 or below will tell me that this particular region is hypomethylated. So that is the values that we get from the microarray, where they actually measure the amount of DNA methylation, the level of DNA methylation, and express in terms of beta values. So that's how we actually study.
Okay.
Okay. I'm
So,
actually I'm only answering in terms of computational aspect because I'm not involved myself in wet lab aspect. Yeah.
so thank you, Dr. Darshan. Since there are no more questions, I
So
really like to extend my deep sense of gratitude to you that even if it is 12 in the midnight, you had a patience to answer to all the questions that popped out in the chat box or YouTube chat box. So, guys, this is Dr. Darshan, whom I really have a high regard as my first batch student. And and he always I quote, as I said earlier, he is the one who always I quote as an example that you don't need to know everything before you go into pursue the research career. Only thing you have to have is a zeal and a mind to give everything into whatever the work that you are doing. So I thank you, Darshan, for the wonderful talk. Hope it inspire our students to take or pursue a research career further. So from all of us from Department of Biotechnology and Bioinformatics, I extend a very heartfelt thanks to you. So
pleasure,
hope
sir. Pleasure.
you contribute much more to the science in future also.
Pleasure, sir. It was absolutely pleasure talking to all. I hope I have uh, able I have been able to share some of my knowledge with you all. And one thing that I would like to mention here is you're all in very good hands, and try to develop that. I mean, try to gather as much information when you are actually studying it. And also, as I was mentioning to Santosh sir in the previous meeting, that one thing that you all should develop is ability to self learn also, because that is what really helps you once you come out of your educational setup. Because wherever you go, whether you go to an academic setup or to the companies or for internship or wherever, the ability to self learn yourself and keep moving ahead will actually take you to the next level and help you succeed in your life in whichever the way that you have imagined, and can open lot of opportunities which you never thought when you're doing the work. So as long as you are dedicated and just have open mind to learn things by yourself, because here you have wonderful teachers and all to tell you, but as you move ahead, there'll be not many people to actually help you. So one has to learn by themselves and get ahead. That's how it should work. Yeah. Okay.
Thank you.
Thank
Thank
you.
you,
Thank you so
Darshan.
much. You're
Thank
welcome,
you
sir.
very
Thank
much.
you.
Thank you.
Yeah.